Welcome back friends. In this class we will learn about tumor immunology. But in the beginning I want to give a special thanks to Farhan and Parveen who have requested me to make this video. Tumor immunology means how our immune system responds when it detects any tumor cells. In this class I will not talk about the molecular reasons behind the development of cancer or tumor cells, because a normal cell actually transforms into tumor cells. So I will not talk about the molecular mechanism behind the development of cancer. And you have to understand one fact, that cancer is not a direct disease. It is actually a complex. It has many facets. So basically this is a wonder. When we learn biology or life science, we actually feel wonder. Why? The reason is because a cell is like a organism that can sustain its life itself, and a subtle balance is present in a cell to sustain its life. So the name of these components of our body called cell is very small. It has only 4 letters, CELL, but the complexity it has is tremendous. You can't imagine. We researchers or those who are actually working on, those famous scientists, also do not understand what is cell, because the complexity that is present in our universe is same or more than complexity is present in a tiny cell. Many things occurs inside a cell. If you start to learn cell biology, you can understand. The cells have its metabolism. Its cells have cell cycle. It has own death mechanism called apoptosis, and growth factors, signalings, many things. It has some movements. It has actin, microtubules, microfilaments, and many things like transportations. So a cell actually keep balance of all these mechanism to sustain its life. So a cell is very complex. So to become a tumor cells, it is not actually a very unusual thing. It is very common thing should be, but we wonder why a cell actually remains as long as normal, because to maintain such a complex work, it should have some intelligence that we till now have not found, or have not find what are the intelligences actually working on a cell. How these cells, tiny cells, can manage to perform all its functions. Okay, so in this class we will not go into the details of molecular mechanism how cancer develops, and there are many nomenclatures. You have to these are called terminology that are specially go for cancer, like benign tumor, malignant tumor, metastasis, and there are many different type of cancers can occur, like we call carcinoma, sarcoma, lymphoma, leukemia, and then other is myeloma, melanoma. There are different type of cancers can be present. These are depends on the tissue, what type of tissue actually go through or went into a transform into a cancer cells or tumor cells. Like if epithelial cells transform into tumor cells, we call carcinoma. If connective tissue cells like bone, blood becomes bone or chondroid tissues, if it becomes into tumor cell, we call sarcoma. Another is blastoma. If embryonic tissue become cancerous, we call blastoma. Lymphoma means if our lymph gland become cancerous, we call lymphoma. Leukemia means if blood becomes cancerous, we call leukemia. And there are different type of leukemia also present. Acute leukemia, chronic leukemia, myeloid leukemia, lymphocytic leukemia. There are many different type of cancers can be present, because we have many different type of tissues. So to make a video on cancer is difficult in a single lecture. So I will not go into details of molecular mechanism, because I will talk about this in another lecture when I will give a cell biology class or molecular biology class. In this class we will learn about the role of immune system during tumor. So I have write down some 10 points that is very important. This is called Hallmark of Cancers. If you search in internet this just by typing Hallmark of Cancer, you will find these 10 different reasons behind the development of cancer. In 2010 and 2011, subsequently, two professors, Douglas Hanahan and Robert A. Weinberg, two professors actually write a review on a very famous journal called Cell. And this hallmarks, it's called Hallmark of Cancers. This is one of the most cited reviews till date. So Weinberg and Hanahan actually published these 10 cause of cancers. So this is not our part today, but I want to mention you for the beginning, because these are the reasons behind the development of cancer. Then I will go to the immunological part. The first point is why a normal cells become cancerous cells or tumor cells. The main problem, not only it proliferate fast, but there are many different reasons behind the development of cancer. Can you see that the first reason is sustaining proliferative signaling. That means in a cell there is a signaling called proliferative signaling. So a cell should proliferate, because to perform its functions it requires proliferations. And in a theory is present called Heflix Limit that tells us that a cell have 60 to 70 times division power. So a cell can divide 60 to 70 times. This is called Heflix limit. But when a cell cross the limit of Heflix, we call it becomes a cancerous cells. So proliferation is actually normal phenomena. Proliferations, like our intestinal epithelial cells slough off every day, our skin cells slough off every day, and they proliferate each and every time. This is good for our health. But if it sustain its proliferative signaling, then it could cause a cancer. This is one reason, just one reason, not the whole reason for sustaining proliferative signaling. Example, there are many examples are present, but I have given just two examples for the simplicity. Like BRAF, these proteins actually perform its normal function in normal conditions, but when it performs its higher functions, it can cause cancer or tumor conversions. Like this is under the MAP kinase pathway, signaling pathway. Another is P10, this is a phosphatase, and PI3K, that means a kinase enzyme that actually rival. They both are rival because phosphatase and phosphokinase are always a rival, and they actually control a pathway called AKT. This is actually proliferative pathway. So I have already mentioned that a cell actually go through a very subtle balance to sustain its life. Okay, and we think that cancer is actually abnormal. No, actually cancer is normal, because to maintain such a complex balance, it is very difficult. But we don't understand how a cell actually keep balance, and keep balance means homeostasis, keep homeostasis of all these process. We call it networks also. So it is very difficult to understand of us, like we perform research, we cannot understand how a cell can sustain such a very complex process in a very stable way. And there is another reason called evading growth suppressors. Now, I have mentioned that there is always a balance. There is always a tug of war between a cell. One type of proteins or signaling try to proliferate, one type of signaling or proteins try to prevent the proliferations. The same is true here. The evading, like growth suppressors, this is the proliferative signaling that helps to proliferate, and there are some proteins that actually prevent proliferation, we call suppressor. But those cells become converted into tumor cells, they can evade the growth suppressor, like retinoblastoma protein, this is a suppressive protein, and P53 protein, this is also a suppressive protein, and tumor cells actually suppress these two proteins. So it suppresses the suppressor, so it can proliferate. Then resisting cell death. So I have mentioned that a cell actually go through its own death, a mechanism. Okay, this is called programmed cell death. We call apoptosis. So in case of apoptosis, they resist this type of cell death. they do not go through apoptosis. Okay, like BCL2, BCLXL, these two proteins are called anti-apoptotic. They prevent apoptosis. And BAX, BEAM, PUMA, these proteins are called pro-apoptotic, that helps in apoptosis. So in case of cancers, these proteins perform its function normal or in high level, and there is problem in these two proteins because they actually anti-apoptotic, they prevent apoptosis. And so enabling, another example is enabling replicative immortality. So for proliferations, a cell requires genome also, genome. So a cell can duplicate its genome or chromosome by replications. So it should go replications and to an unlimited time, that is called immortality. Cancer cells or tumor cells are immortal cells. And how they become immortal? By an enzyme called telomerase. This telomerase enzyme is very important for normal cells also. But in normal cells, as the cells actually divided gradually, and when it actually go to the Heflix limit after 60 or 60 to 70 times divisions, the amount of telomerase enzyme starts to reduce in the cell. So if you actually know about replication, you must know that during the replication there is a problem called end replication problem, and that problem is solved by telomerase. And what happens, each and every time when a cell divides, the end portion of the chromosome actually shortened for every subsequent divisions. But telomerase helps to cope up this problem, and tumor cells express this telomerase high level, so they can go through this problem and actually they become immortality. So these are all reasons. This is called hallmark. Another is inducing angiogenesis. This is another important, very important fact about cancers or tumors, because when tumor grows, it requires high amount of nutritions, okay, than other normal cells. Other normal cells actually go through a metabolism that have glycolysis and Krebs cycles, but in case of tumor cells, they don't go through Krebs cycles. They only perform glycolysis and produce from glucose to lactate. So they generate very low amount of ATP. So they require high amount of glucose, high amount of nutritions. So they actually secrete some molecules that helps to make many arteries and vesicles, vein, for supply of blood. So they actually help in angiogenesis. Angiogenesis means formation of blood vessels. So when a tumor actually grow, in the surroundings, there are actually different arteries and veins are actually produced. We call angiogenesis. And one example of angiogenesis that actually promote angiogenesis, this is called VEGF. This protein is actually produced excessively, that helps in angiogenesis in tumor cells. And another is activating invasions and metastasis. This is very important term, because when a tumor grow one side, it can actually escape the primary tumor site. It can go into any other organs. Like if anyone have a liver cancer, these liver tumor cells can go into kidney, can also go into brain, can go into heart or liver or other organs. So it can actually migrate. This is a very specific term for cancer called metastasis. Another important term we use called EMT, that means epithelial to mesenchymal transitions. The cells that is epithelial in nature become mesenchymal in nature. It can migrate like an amoeba. So amoeba, just like an amoeba move, these cells can move. And there are some so there are many things to talk about these tumor cells or tumor cells specifically, that in case of normal cells, when we culture normal cells, the cells actually can sense that if there is huge amount of cells are proliferating, they actually stop their proliferations. This is called contact dependent inhibitions. When in the culture plate the number of cells actually proliferate and totally cover the plate, culture plate, then it stops its proliferation. It's called contact dependent proliferation. But tumor cells do not go through this contact dependent proliferations. Somehow this mechanism actually disrupted. Another is, we see that for a normal growth of cells, the cell must actually attach with its basement membrane. A cell must attach with the basement membrane. This is called attachment dependence. Dependence. But tumor cells can actually survive without attachment with the membrane, so it can actually migrate. And it is called invasions also, because it invade. And some proteins are actually responsible for these invasions, like NF2, neurofibromatosis, and 3 proteins, that is snail, slug and twist, are very important for this metastasis. And there are many different proteins are also responsible, like cadherin proteins. There are some signalings also responsible, like TGF beta signaling. So these are all actually topic. If you want to learn more about this topic, you can learn. There are many things to learn. Okay, and I will refer you a book for specifically cancer. If you want to learn about cancer very specifically, just one book is sufficient. The book name is Cancer Principle and Practice of Oncology by Rosenberg. I also recommend this book, and I read this book, and it is the 11th editions, Cancer Principle and Practice of Oncology by Rosenberg, 11th edition. This is recent edition 2019. I also read this book, but the problem is that this book is so it has so large volume. It has 4,300 pages and 47 chapters, single book. So this book have all informations about cancers, recent discoveries of cancers, because this topic is actually under research. It is not actually completed. We don't know about cancer totally. Maybe we know about 2 to 3%, 97% we don't know. So another is genome instability and mutations, that is new antigens. New antigens means if there is any mutation occur, we call it new antigens. So that type of protein actually cause genome instability, okay, and mutations. Deregulating cellular energetics. This is also very recent research. Scientists have found that those cells become tumor cells. They actually go through a different metabolism mechanism. And another one example just I am giving, a molecule called CD36 is also responsible for this mechanism. Another is tumor promoting inflammations. This is very unusual. This is actually under the tumor immunology, because we think that our immune system always try to kill tumor cells, but sometimes it not. Like you can see here, inflammation can cause, like tumor promoting this type of inflammations. So our immune system has a dark side, so it can promote tumors, that like example is necrosis. And there is another tumor related reasons behind the development of cancer is avoiding immune destructions. Tumor cells actually gain a type of protections or mechanism by which it can avoid immune destructions by producing high level of T regulatory cell, and there are some suppressive molecules are present. I am just giving some examples like CTLA4 and PD1, but there are a huge amount of molecules are actually responsible. We are working on this topic. This is very recent topic. This is called immunotherapy. We are working on immunotherapy. There are very in backward time, there are two therapy are actually present, like radiotherapy and chemotherapy. Some drugs that are actually we doctor treat, but now it is emerging. This therapy is emerging called immunotherapy. Immunotherapy. That means we use our immune system, we boost our immune system, or we manipulate our immune system to cope up, to kill the tumor cells, not with any medicines or chemical, but with our immune system. So this is called immunotherapy. This is very recent therapy, and the positive side of this therapy is that it has very low or negligible side effect. Okay, but also it has some side effect, and there are some problems in case of immunotherapy. I will talk about this. So these are the hallmark of tumor cells. Now I am not going into more details because I will talk about this in my cell biology part, when I will give more details, because these are all individual topic that can I actually give lecture. So I will not go to details, but you should learn about these 10 reasons. This is called hallmark of cancer. Must search in the website, you will find the diagram that is actually used in the review paper. So yes, so yes, there are some molecular mechanism behind the development of cancers also, like virus can cause cancers. There are some virus like HPV virus, human papilloma virus, HTLV virus, human lymphocyte T cell virus, and there are Epstein-Barr virus, and hepatitis B virus, SV40 virus, simian virus. So there are many different viruses are responsible for generation of tumor also, because they have some gene, or they can transform our genome. It is called oncogene, or we call it viral oncogene. Because cells also have some oncogene, we call proto-oncogene, that is not harmful, but when it becomes into aggressive or bad side, we call it viral oncogene or oncogene simply. Now, so where is the role of immune system? Because these are the role of, or these are the cause of development of cancers. A cell go through a complex mechanism, so there may be few problems can occur. This is actually normal. A cell is normal. This is actually very abnormal thing, because there are so many circuits. If you look in internet, you can find that a single protein is actually related with thousand and thousand proteins also. So those actually working on these cells, they know that this is actually nothing but a circuit, like electric circuit, like a mobile chip or computer chip. Okay, so everything is related, not a single protein is working its own accord. So now I will talk about the role of our immune system, how immune system actually detect tumor cells. The problem here is our immune system are trained in such a way that they can recognize foreign antigen. This is very interesting. Our immune system are trained to actually detect foreign pathogen, like specifically foreign pathogen, like bacteria, virus antigen, fungi antigen, protozoa, parasites. They actually recognize efficiently this type of foreign antigen or pathogen. But our immune cells like T cell and B cells are actually go through a process called self restrictions. I have also taken that type of video or lecture, that they go through a self restriction. That means they do not recognize our cell proteins. And the problem is that our normal cell actually transform into tumor cells. This is right. Our normal cells transform into tumor cells. So there are so many similarities, or so many proteins are actually present in the tumor cell that is also present in the normal cells, because it is nothing but our own cells that actually become our enemy, because our own cells are our friend, but when it transforms into tumor cells, it becomes our enemy. But it has some features or some proteins that are actually shared with the normal proteins, normal cells. So our immune system actually falls in a very difficulties to identify that it is a tumor cells or it is a normal cells. But yes, somehow it can manage to identify few antigens. Okay, these are called tumor antigens. Scientists have found that there are two types of tumor antigens are present. They call it tumor specific antigen. Another is tumor associated antigens. they found that in case of tumor specific antigen, that means it is specifically present in the tumor cells, but some antigens are associated. I will talk about this. So tumor specific antigens are example like viral gene product. So when a virus infect a cell, that cell could be transformed into tumor cells. Example, human papilloma virus. This virus can cause cervical cancer, uterine cancer. This is for actually this virus actually cause cancer in females. So this virus have some specific proteins like E6, E7 that could actually be recognized as tumor antigen. Another virus is HBV. I am just giving two examples. There are many viruses and many antigens are present, but for simplicity I am giving just two examples. Hepatitis B virus, and there is an antigen called hepatitis B surface antigen. This could be also recognized as a tumor antigen, and this cause actually liver cancer. Now there are some new antigens, like I have mentioned here. New antigens means that actually go through mutations and different in normal proteins, like VSRC. This SRC protein or SRC protein is actually present in also our normal cells. This is called CSRC, means cellular SRC. This is actually proto-oncogene, but when it transforms into oncogene we call VSRC, because a virus called Rous sarcoma virus, in the first time one scientist actually discovered Rous sarcoma virus cause cancers, and they actually discovered that this virus have a oncogene called VSRC. Okay, in our normal cells there is a CSRC, means cellular SRC protein, but in case of virus these have a VSRC protein. So another is new antigen like new. This is present. This is actually caused in breast cancer and some ovarian cancer also, but specifically breast cancer. New protein. This is actually a transformed protein. The normal protein is HER2. In case of normal cells, these are called HER2. This is actually a receptor for EGF. That means epithelial growth factor. It is a growth factor receptor, but this new protein is actually a deleted product of HER2. And another is RAS-D. So RAS is also a signaling protein, but in normal cells it has present, but it is a mutational actually a product of RAS protein. In case of RAS, the 12th number of amino acid is glycines. If glycine is changed into any amino acid, it becomes it actually helps in proliferations and uncontrolled proliferation, because RAS protein actually helps in proliferation, but in a controllable process, because RAS can be activated and deactivated. So in our cells that the proteins are present that could be switched on and switched off, but this type of proteins cannot be switched off. They are always on in conditions, like this RAS-D product is always on in conditions. Okay, now so these are the tumor specific antigens that are present in tumor. So immune cell can very easily detect this type of protein, but there are some proteins are also present. These are also present in case of normal proteins. But how our immune system can recognize this type of antigen? One is called location. Like you can see here, a type of antigen called cancer testis antigen. Under this family, 200 proteins are actually there. I have not written any of them because you don't have to remember the name. Just remember the type of function. Like the location can be identified as a not as normal, because this type of antigen is present in gamete or testis. But during tumor transformations, when normal cells become tumor cell, then their transcription regulation is not perfect. They can synthesize this type of abnormal antigen that are not present in normal cells, not present in liver cells, but when liver cells transform into tumor cells, that also can produce this type of antigen that is present in testis or a gamete that is abnormal. The immune system can recognize this abnormality. Now this is location. Another is timing. Timing can be actually detected. Like this type of proteins are called oncofetal. The term actually denotes that this is onco means tumor specific, Onco means tumor or cancer. Fetal means fetus. So these type of proteins are actually present in during the fetus time. Okay, so it should not be present in normal or adult time. So AFP means alpha fetoprotein and CEA carcinoembryonic antigen. These type of proteins are not present in normal individual but present in 2 to 6 month fetus. When a fetus, human fetus, the age of this fetus is 2 to 6 months only in the mother's womb, then these proteins can be expressed, and that time actually the baby do not develop immune system. So immune system do not recognize this type of proteins. So if any tumor cells express this type of protein, they can recognize that this is not our own cells. Okay, another is level of expressions. So you can understand these proteins are actually normal, but the timing, the location is different. That's why immune cells actually recognize it as a tumor associated antigen. Another is level of expressions. This is called like CA125, very commonly used in case of ovarian and uterus cancer. This is actually a glycoprotein. It can be expressed by the cells but low amount. But when it becomes cancerous or tumors, they can synthesize more vigorously or high level. But I want to mention that this is actually performed test, but this is not very conclusive, because when a woman go through a menstruations, this type of antigen can express highly. So this is not true to just test CA125 and you can confirm that she has a cancer. No, you can get an idea that maybe she has some ovarian fibroids or some uterine fibroid. Like PMSA, prostate membrane specific antigen, there are other prostate specific antigens also present. So another one example is PMSA, prostate specific antigen. This is also present by prostate cells but in a low amount or in a moderate amount, but in case of prostate cancer this protein is expressed highly. And there is tyrosinase, this is also the same too. So this is the beginning of our immune system. But how our immune system react when it detect immune or tumor cells, that is the topic of this tumor immunology. So I will show you. So you must search the name I have mentioned, that is Hallmark of Cancer. This is very important. Hallmark of Cancer. This is one of the most cited journals or reviews till date. So now I will talk about how our immune system react. Okay, this is very important because there are some cells are present that actually respond. So this is called, when our immune system respond towards a tumor, we call immunosurveillance. This is one term, immunosurveillance, but another term is called immuno editing. Immuno editing. So I have also recommend you a book by Rosenberg. You can also learn this book, Cancer Practice, Principle and Practice of Oncology. But the volume of the book is so high, 4200 pages, but you can learn as you want. Okay, so there are so many things about tumors or tumor immunology and tumor cells. It is very difficult to make you understand by a single lecture. It is very difficult, but I am trying my best. Immuno editing, this is another term. Under this term there are 3 E. You can remember this. 3 E are present. E means elimination, equilibrium and escape. We use this 3 E. Must remember this. This is very important for tumor immunology. Many teachers, many professors and many doctors very unconsciously say that a cancer is a very rapidly proliferating cell or tumor is a very rapidly proliferating cell. But this is not actually true in a broad sense or in a larger sense. This is not true. I will explain you. Immuno editing, under the immuno editing, that means our immune system are actually working, because in our body immune system are always working. So let's assume if there is a transformation of a normal cells into a tumor cells, what happens? Our immune system immediately try to eliminate that type of tumor cells because they can recognize tumor antigens. A tumor normal cells, when become tumor cells, it starts to express this type of antigen, and this type of antigen can be recognized by immune cells like T cell and B cell or antibody. So our immune system try to eliminate the tumor cells. The first thing is that, but we know that there are cancer patients. That means our immune system fail. So our immune system actually fail, and that leads to the second phase, that is called equilibrium phase. What happens? Why we call equilibrium? Because in this phase, in that phase, there is no war between immune cells and tumor cells. That is very interesting fact. This is also called, this equilibrium is also called dormancy. Do you know what does mean by dormancy? That means just sleeping. Tumor cells do not go to fight. They actually wait for the time when our immune system will be weak. Let's assume if any persons have any infections, have any heart problem or any immune system problem, if his or her immune system actually down, then that time this cells that has actually become tumor cells, that can actually get the opportunity to proliferate. This is called immune equilibrium. So when a patient actually develop tumor cells at the beginning, but when the cancer actually produce, that can take approximately 1 year to 10 years. Can you imagine? Now you cannot say that tumor cells grow very fast. No. Tumor cells proliferate very fast in the third phase. You can see here, escape phase. But at the first, immune cells and tumor cells fight with each other. These are called elimination phase. And sometimes immune system win, sometimes tumor cells win. If immune system wins, that person will not have cancer. But if the tumor cells win, then it will not directly go to another fight. It will wait. Few cells, it may be 1,000 cells, 1 lakh cells. Don't go to the number because cells are so tiny that 1 lakh cells also produce a seed-like mass. Okay, so at that time they go to dormancy, and the dormancy can take 1 to 10 years. It can take longer times, and it depends on the person's immune systems, how the immune system react. This is very important time, because I have seen many patients that go through uterine surgery or ovarian surgery, and doctor have actually thought that she can have, or actually in case of females I have found that they thought that she have cancers because it have taken so many times. But after USG they found that there is no cancer, so they can go through surgery. So that depends on the immune system of the person. So equilibrium phase is the longest phase in case of cancer development. Now when our immune system actually low, then these tumor cells escape, then it actually come into the surface, then it proliferate first and cause cancer, because tumor cells then metastasize. It actually go out from the primary center to the whole body and spread all over the body. It actually break the all system. It actually change the total body fluid nature. If you check the blood pH or any substance, you will find that there are difference in normal and cancer patients, because it all totally change in this escape time. Totally the body physiology actually change and actually hijacked by the tumor cells. Now this is the immune functions. Now I will show you a experiment by which scientists have understood that immune system have role. They perform a simple experiment. They took wild type mice. They also took a RAC2 knockout mice. Okay, so wild type mice means, you know, that is natural mice, and RAC2 knockout mice, RAC2 means it is actually a enzyme that helps in diversity. I have made a separate video on class switching, and in that class I have mentioned what is the role of RAC2, class switching. And class switching is a very important mechanism for the diversity of T cell receptor and B cell receptor. And as much as the T cell receptor and B cell receptor are diverse, they can recognize diverse amount of antigens. So RAC2 knockout mice means that, knockout means, if you know about knockout, but I want to mention here what is knockout. Knockout means the gene is actually deleted out from the beginning, from the zygote. Okay, so this RAC2 knockout mice means these mice don't have RAC2, and RAC2 is actually beneficial for our immune system. So those mice don't have RAC2, we call immunodeficient. Immunodeficient. Okay, so these immunodeficient mice, they actually injected. We can grow tumors in mice. We perform this experiment. We inject with cell line. Okay, like in case of breast tumor we inject 41. In case of melanoma we inject B16. Okay, so scientists actually grow tumor. They inject tumor cells and grow tumor in these two type of mice, and they then take a wild type mice. They also take wild type mice. They take wild type mice and they transfer the tumor from this wild type mice to these wild type mice, and from the RAC2 knockout mice to the wild type mice. You have to understand this. But yes, I have to mention here, this wild type mice is actually syngenic. You have to understand this. Why it is syngenic? These mice are syngenic. And what does it mean by syngenic? Syngenic means genetically identical. You have to understand one fact, that when any tissue, I have mentioned in my transplantation immunology class, that if anyone transfer tissue graft from one species to another species, it will reject. Not only that, if you transfer tissue from one individual to another individual of the same species, like in a human, if you transfer liver from one liver, not because liver is not highly immunogenic, but like kidney, if you transfer kidney from one individual to another individual, it will reject. So genetically different individual can reject a tissue. So for that reason, scientists have taken genetically identical mice, we call syngenic, so that it will not reject the tissue. So when they transfer tumor from the wild type mice to the wild type mice, syngenic mice, they found two very interesting facts, that in this mice they show aggressive tumor. So they took wild type mice, they grow tumor, and they transfer or transplant the tumor into the wild type mice, that is normal. They found that this tumor actually grow aggressively in these mice, but when they transfer tumor from RAC2 knockout mice to the wild type mice, same mice, they found approximately 40% of the mice don't develop tumor. Now this is very interesting. So what happens in this case? A simple thing happens, that is called, very interesting, this Darwin is not only restricted to evolutions or ecology, it is also restricted to immune system, not only immune system, in our body physiology also. Darwinian selection has occurred. and that is the phase of this elimination phase. In this elimination phase, this Darwinian selection occurs. I will talk about this in details. Here you can see in a wild type mice where the immune system is just sufficient because it is not immunodeficient. In case of wild type mice, the immune system is sufficient. So when immune system is just enough, and when this type of mice grow tumor, that means the immune system, that is actually active immune system, try to eliminate tumor cells. Okay, but I have told you this is a tug of war. This is a war. So a immune system can kill all the tumor cells, but somehow some tumor cells can escape or have the ability to escape the killing by the immune system. So in wild type mice there is a activating immune system that actually try to kill the tumor cells, but it cannot perform it totally. So those tumor cells actually grow under the activating immune system. They are less immunogenic. You have to understand this term. These tumors that grow into the active immune system, they are less immunogenic. That means they cannot be identified easily by immune system because they have survived. They have go through this Darwinian selection process. There is always a selection between these tumor cells. Those tumor cells are less immunogenic. They are selected. They cannot be killed by this immune system of this wild type mice. So when this type of tumor, that are actually less immunogenic, are transferred into wild type mice, they progress aggressively. They cause aggressive tumor. But in case of immunodeficient mice, you have to understand this. Immunodeficient mice, where the immune system is not properly function, not can kill, not have diverse receptors. What happens? That type of tumor cells is more immunogenic because it has not go through selection process. So you have to understand this. If your batchmate are more strong, more studious, then you have to work hard. This is very simple thing. If you have a batchmate, your classmates are very studious persons, then you are gathering through these persons, then what happens? You have to study well to cope up. That's also true. And let's assume if you have a very weak classmate and you are the actually winner or king of that classmate, then you will not sufficiently prepare for yourself. Okay, so that happens actually in case of immunodeficient mice. Immunodeficient mice have low immunity, so they do not go through this Darwinian selection process, so they are more immunogenic. So those tumors are more immunogenic, that can be recognized by immune system. So 40% mice don't develop tumors because they can kill that type of tumor cells because they do not go through this Darwinian selection mechanism. So from that experiment, scientists come to know that yes, our immune system actually not always good for our tumor progression, but it is also can be bad for immune system. So yes, now I have to give you some example of immune cells that actually responsible for tumor immunity. The examples are like, the first thing that come into my mind, this is the NK cell. I will not talk about how NK cells kill tumor cells because I have made separate video on NK cells. NK cells is, the name implies natural killer cells. The name implies that it is born to kill, so it can kill tumor cells and viral infected cells. It has some mechanism. I have discussed about this topic in my another lecture called clever NK cell. I have made a separate video that heading is clever NK cells. You can watch that video. So NK cells actually one of the soldier of our immune system that eliminate tumor cells. Another, we can group these cells as a good inflammation and bad inflammations. inflammation means immune system or immune cells. You can understood as like this. So in case of good inflammation that actually helps in the elimination of cancer, the example of cells are M1 macrophage. And what is M1 macrophage? You have to see my Th1 and Th2 cell development. Then there is cells like dendritic cells. They can perform a good role in to eliminate tumor cells, but these cells should be cross-presenting. That means dendritic cells generally express its antigen through MHC2 molecule, but in this case, to clear the tumor cells or to kill the tumor cells, it must represent its antigen, tumor antigen, through MHC1 molecules, so that CD8 cells can be activated. So this dendritic cell activates CD8 cells. CD8 cells are called cytotoxic T lymphocytes. That means this is also a toxic cells. It can kill directly T cells. And also this macrophage, M1 macrophage, activate a type of helper cells. This is called CD4 cells, actually CD4 positive. This is called Th1 cells. I have made separate video on this topic because so I cannot talk about this in more details. Just I am giving you the example. Another is in case of bad inflammations, I am making it in red color so you can distinguish. In case of bad inflammation, there is another type of macrophage is actually involved, that is called M2 macrophage. You can understand what is M1 and M2 macrophage if you watch my Th1 and Th2 video. So this is M2 macrophage, and there is a called MDSC, myeloid derived suppressor cell. So this is another cells. This is suppressive cells in our immune system. So those cells are suppressive in nature. They help in tumor growth, like Treg cells also helps in tumor development. So now you can see, and these cells can secrete different cytokines called anti-inflammatory cytokines. Like these T regulatory cells, we work on these cells. They can secrete IL-10, TGF beta, IL-35, and many things can occur. These cells express high amount of CTLA4, a suppressive molecules. So that actually cause that actually when bind with this ligand, it actually activates an enzyme called IDO. That intolamine deoxygenase enzyme can activate or break tryptophan amino acid into chitreorenin. That chitreorenin is actually suppressive in nature. So there are so many knowledge, so many informations are actually there if you want to learn about tumors or immunology. But we actually perform, those who work on immunology or immunotherapy, we don't actually learn all these things. Just I am working on a very particular molecule called CTLA4 and PD1. And in very recent years, 2018, two professors, very eminent immunologists from America and one from Japan, the American scientist called J.P. Allison and the Japanese scientist Professor Takasu Honjo, were rewarded Nobel Prize in 2018, just recently, for the discovery of monoclonal antibody against CTLA4 and PD1. I am working also this molecule with another aspect. So I have to write something about this. So these are the cells that actually helps in this process, but there are some cytokines that are also responsible for the elimination of tumors, like IFN gamma, IFN alpha and beta. So these IFNs are very important. This is called type 2 interferon. This is called type 1 interferon. So this 2 interferon, 3 interferon actually very important for elimination of tumors. Another type of interferon called TNF alpha. That means tumor necrosis factor. This the name implies this is against the tumor. And another is called IL-12. Yes, another cell I have forgot to mention in case of bad inflammations, we have also found in our laboratory that Th2 cells. So this M2 macrophage, Treg cells, MDSC and Th2 cells actually cause poor prognosis. They cause good prognosis, they cause poor prognosis. So as I have mentioned in my last point, I have mentioned that tumor cells can avoid immune destructions. Tumor cells have some mechanism that can avoid immune destructions. So we are working on this type of system. We call it immunotherapy. So yes, I want to give you, so these are the mechanism. Like NK cell can perform ADCC, can perform perforin and granzyme destructions. This macrophage can engulf. So they perform different functions. CTLs can directly kill tumor cells. So if you want to learn about this all cells function, then you have to learn in more details about these cells. Okay, so now I will show you some therapy that are used as a immunotherapy, because I will not go into chemotherapy or radiotherapy. I will talk about this in my cell biology class, but in tumor immunology class, I will talk about tumor specific immunotherapy. So we will learn tumor immunotherapy. There are many immunotherapy are present. FDA, the American actually agency, approved this type of drug. So examples, I will give you few examples that are very famous, like rituximab. We also call MAB. You will found that this MAB full form is monoclonal antibody. Always you found rituximab. I will show you another is ipilimumab. This also MAB. Another is like nivolumab. There is another thing I have not mentioned here, that is called trastuzumab. I am not mentioning here because this is not under the also not immunotherapy, because it do not target the immune cells or immune receptors, because trastuzumab is actually target this new protein. So I will not discuss about this. So I will show you what is this monoclonal antibody against. So this monoclonal antibody, this is actually antibody. These are the antibody that actually against a molecule that is present in cells, immune cells, like CD20. This is against the CD20 molecule that is present in B cells. So in case of B cell cancer, so in case of B cells leukemia or B cell cancers, the CD20 target actually performed. And ipilimumab is against the CTLA4. In 2018, two scientists actually rewarded Nobel Prize for these two antibody, monoclonal antibody. Nivolumab for PD1. This is another suppressive molecules. So this antibody can bind with this molecule and can inhibit the inhibition process of this two molecule. So yes, another thing is vaccinations. Vaccination can also be performed for immunotherapy, like the first approved vaccine is called Sipuleucel T. This is also a dendritic cell mediated vaccines also approved, and there are many different things can be performed, like T cell adaptive transfer, and another is called CAR-T. These are the very recent immunotherapy. And I have mentioned a vaccine called Sipuleucel T that is used as a dendritic cell vaccines. If you learn about this vaccine, you will found very awful that by the treatment cost of this Sipuleucel T vaccine is actually approximately $1 lakh in US. In our Indian price, it could be 1 crore. But the survival longevity, when you treat with these vaccines, the patient just survive for 4 months extra. Can you imagine? The patient survive 4 months extra and you have to pay 1 crore for that. And this therapy are so all is personal therapy called T cell adaptive transfer. These are the common therapy. This could be performed in many individuals, but these have some side effects. But these two therapy are so costly that it is called personal therapy. A person have cancer, the total scientist group and total medical group will work for a single person. So it will take approximately 10 crore or 20 crore. It may be for the total therapy. This is called personal therapy, T cell adaptive transfer and CAR-T. If you want to learn about this, you can watch. You can actually search in internet And because in one class I cannot mention all these things in more details, I have also recommended you a very good book, Rosenberg. So hope this video will help you. Thank you for watching, and always, that is very important, keep learning.